So I'm Dan Cox, and on behalf of the Dean's Office, I'm happy to welcome you this morning to a discussion on core facilities. Um, we have three excellent panelists, John Houghton in biology, Chai Chen for the imaging core facility, and Ximing Wang for the chemistry core facility. And each of them is going to present on various instrumentation capabilities, services, whatever they would like to highlight around their core facilities. And also from the group that's tuned in, we certainly welcome feedback on maybe instrumentation needs that you have that we don't currently have access to, because thinking forward about how we empower the faculty to do the most cutting edge research is certainly important as well. So I thought I would start with Ximing. If you want to.
Okay.
Everyone can be presented just for.
Okay. First of all, let me just share my slides with you. Hold on a second. Let's say share the screen. Can you all see my slides?
Not yet.
Not yet? All right. So let me just try this again. I should be able to do that because I'm teaching with WebEx. So here you go. All right. Now can everybody see it?
Yeah.
Good. All right. Well, first I'd like to introduce myself. I'm Ximing Wang. I manage the mass spectrometry facility in GSU. Actually, it's not the core facility for everything. So I'm only managing core facility for mass spectrometry. Well, first of all, I would just like to show you my contact information. This is the website of mass spec facility, and you will find the information from this website. And also, this is my lab numbers, and just because the instrument I have spread out over different labs, so the best way to find me or contact me is through my email. This is my email address, and Lenore Allison is my assistant, and this is her email address. So in case everybody has any question regard to the service, regard to the facility, and that's the best way to contact me. All right, so I will just briefly introduce the website we have, and the website of mass spectrometry actually lists most of the information needed in terms of the instruments we have, the applications, and a description of the instrument, and also the protocols and the useful links. That's most for student. You would find out how to predict the fragments of, let's say, fragments of peptides or proteins, and how to calculate, the exact mass of different molecules. That's just have a lot of information and useful links and how to do so. And also,
Ximing, just can you switch to the presenter mode? It would just
that's
bigger.
Sorry.
No problem.
Okay, here you go. This is probably better. I can see better. All right, so we also have one tab is just for instruction how to submit the samples. The instruction how to prepare the samples, how to submit the samples, which also include the form which is needed, along with the submission of the samples. Well, just, for the general information of mass spectrometry facility in terms of service and research collaborations. And for the internal, we open to everyone on campus, and with just, internal charge, the charge is the same to everybody on campus. For external, we also provide service to Georgia, all Georgia academic communities. We all just share the information, share the instrument, and we also charge the same rate, the same on-campus rate. Okay. For the turnover time, and after you submit the samples, and we have 1 to 3 business days to have the data back to you. But often, if you need immediate, requirement for the analysis, just contact us in advance, and we'll just, work with you and set up the time just for the time of your need. For sometimes, if you have special need, you want to just monitor, specifically monitor your reaction or specifically monitor the type of molecules you want to see, and we always just, work with you and just work with you side by side. Now, maybe it's not the best time to work side by side, but it's just, what we offer. For research collaborations, we have been, collaborating with different, with our faculties at different departments, working on the different projects. So often we just, sit together and you let me know what problem you have, you want to solve, and we'll see how to use the mass spec instrument technique to solve the problem in your research. And also we help to develop method, to do the method development, and especially for quantitative analysis. So because you need to find out specific internal standard, how to just set up the sample preparations, and how to provide the quantitative, how to provide the samples. So we can just help you to develop the method to generate the standard curve, especially for the quantitative analysis. For qualitative analysis, it's you just let me know what do you want to know from our analysis. Based on the information you need, we can just help design the different experiments and with different type of instruments. That's currently the 5 instruments we have. Like what I said, I have 5 instruments. It's located in the 5 labs. This one is the oldest instrument we have now, and it's Waters Q-TOF micro mass spectrometry, and which has electrospray ionization source and Q-TOF analyzers. Different instrument has different ionization source and analyzers, which just provide a different type of information. So that's really depend on the type of molecules you want to analyze, and also depend on the information you need from those analysis. We have to choose different instrument. Okay, so we have to probably work with you on the sample preparation, how to prepare those samples. That's really depend on the type of molecules. So for example, if you have very small volatile molecules, we have to choose like GCMS to do that analysis. And the 5 instruments we have, again, is with different instruments that we have to provide different informations. For general, this one, or just briefly introduce the 5 instruments we have. And this one is Waters Q-TOF Micro. It's 16 years old instrument. And that's GCMS, which is we just inherited from Coca-Cola last year. And they're still, they're going. Well, last year it was trying to help us to set up these things, but pandemic things came, so we have not been able to finish that. They're going to come back to finish the installation of this instrument and test of this instrument before we can fully use it. And this one is the this one is the SciEx API 3200 triple quadruple instrument, which couples with HPLC. And this one is mostly just used for quantitative analysis. So I have my own grant, and I work with people from University of Virginia. So we work on the detection and analysis of some neural transmitters. We use this to do the quantitative analysis. I also work with people in biology, nutrition department and in chemistry, and also quantitatively analyze small and large molecules with this instrument. So this instrument, those two instruments are just, our old instrument. That's just, we purchased that with startup package from Bing Wang in chemistry. And these two instruments, okay, this one is Fisher Scientific LTQ Orbitrap Elite, which has the most, the best resolution and accuracy, which is mostly used for proteomics analysis, protein identification. And so because it couples with Cadbury LC or Nano LC, it also has nano ionization source, which is used for very small amount of samples, most of it just, protein samples. And this is the Bruker Ultra Flexstream multi-TOF instrument. So that's, is 7-8 years old. And both of these instruments from start package of George Wang, who was the faculty in chemistry department. And that's from his startup package. So those are just, relatively new, but it's also close to 10 years old instrument. So this multi-instrument most work with, large identification or characterization of large molecules like, DNAs, proteins, lipids, and nanoparticles. So we have worked, on those samples with this instrument. Okay. Just, now we're facing the challenges how to replace those instruments because, they're just where it's aging to the time and we, have to replace that. And luckily have worked 3 years on the instrument proposal from NIH, and luckily last year I get the proposal funded. So we're in the process of the purchase a new instrument which is going to replace this instrument. Okay, so this one is also from Waters. It's Ziva G2XS electrospray QTOF instrument. So essentially, it's just replace this one. It also has QTOF analyzers, but it couples with UPLC, which we can use that for routine analysis of small molecule characterization, protein characterization, and DNA characterization. At the same time, we can also use it for proteomics analysis of, for protein IDs or quantitative analysis of proteins. So hopefully, and this one will be installed probably in March. All right, so for general service and applications, there's just, several type of analysis or services we do every day. For the, characterization of molecules obtained from different sources. And so for basic mass determination, and we characterize small molecules from synthetic source or proteins, nucleotides, lipids, reaction mixtures, metabolites, or complexes. So that's just, routine analysis for characterization of mass from different sources and ranging from small or very large molecules. And also we do elemental composition analysis. That's just help to characterize unknown molecules with their chemical composition. We also do structure and conformation characterizations. That's most time for large molecules. For small molecules, we can analyze fragments and to provide information in terms of fingerprint of structures. For large molecules, especially for protein, we can do protein identification and post-translational modification studies, and also molecular interactions, which includes, DNA ligand interactions, protein, small molecules interactions, protein-protein interactions, protein DNA interactions. We have done all those type of analysis before, so they work really well. Another, major part of service or collaboration is the quantitative analysis with our LCMS instrument, which used to monitor synthetic or biological reactions, or monitor the biomarkers. Currently, we have been collaborating with 3 groups using this instrument to do the quantitative analysis of different type of molecules from different type of sources. That's just the general information. Again, so if you need specific information or about the instrument, about service, about collaborations, and you know my number, and you can just always contact me. Then I'm just going to stop here. So and if you need more detailed information, I have slides for the details. But I think we'll just have general information first. Thank you.
Thank you. And if the panelists are willing to share their slide decks, we can make those also available, since I know I'm taking notes quickly, but I may not catch everything. Are there any questions for Siming? Maurice?
Hi, Siming. Thank you very much. I was curious about 2 things. One, you talked about doing HPLC, that's old school, for neurotransmitters. And I was curious what neurotransmitters you were quantifying. And I wanted to know a little bit more about your internal rates. Can't hear you.
Muted.
Yeah, you're muted.
You're on mute.
If I had $1 for every time I was told that.
It's probably Dan put her on mute. It's all Dan's fault.
Okay, all right. Okay, I muted. Can you hear me now?
Yes.
So for the neurotransmitters, we worked with dopamine, 5-HT before, and I'm currently working with somebody on creating ADPs. That's not neurotransmitters, but something in brain where extract those things from brain samples and analyze the quantitatively analyze ATPs, creatine phosphates, and also worked on the amino acids analysis, quantitative analysis of amino acids level from
Like
I think from I forgot from which tissue, and this is from somebody in biology. Yeah.
is when you say amino acids, do you mean like GABA and glutamate, or?
No, I mean amino acid is just amino acids. So
Yeah.
I think from Dr. Liu's group in biology, I think we have analyzed, worked with them on analyzing amino acid levels. I forgot from which tissues. Yeah.
And you're measuring indices of energy like ATP as well.
We just analyze the level of ATP, okay, at different conditions. Okay, so people from Virginia and their work on the biology part, and they just provide the samples. Okay, so those samples just contains ATP at a different quantity just because of what they have used to treat the animals. Yeah.
Wonderful, thank you. So what are your rates? Does it I guess it depends on the piece of equipment and.
No, the rate here is very generous. And when I start work here, that's back to 17 years ago, and the rate is still same. But we're probably looking to change it after the new instrument is installed. But right now it's $8 per sample for just regular analysis. For high resolution analysis is $10 per sample. And for LCMS is $16 per sample. But if I do the method development, and that's just $60 per hour.
$6 an hour? Is that what you said?
$60.
Okay, that makes more sense. Thank you.
You're welcome.
I had a quick question on, I imagine the Orbitrap would be the most sensitive, and is this detection down to pico or femto?
Pico is very easy to get.
Okay.
But it's also dependent on the size of the molecule. So the larger the size of the molecule is, the sensitivity drops.
Right.
Yeah,
Right.
but most of the time when we work with, protein samples, we have to digest it into peptides. So that will just bring the large molecules into much smaller fragments. So that's peptide, most of the time we're seeing 2000 molecular weight.
Okay, great. Thank you.
Congrats on the grant.
Yeah, that's awesome.
Hi, Siming. Thank you for the presentation. I have a couple of questions too.
Sure.
So to build on what Maurice was asking, do you do also peptide hormones quantification? Like
Yes.
NPY? Okay.
Yeah, the peptides, as long as the most important part for quantitative determination is the detection limit. So, we have to use the standard molecules to find out what is the detection limit with our instrument. So then if that meets your requirement, yeah, that's fine. And
Okay.
another thing is for quantitative analysis, most of the time we need standard molecule to generate a standard curve, be able to, quantitatively analyze it. But for peptide, so as long as you can have the standard peptide, we can use that for standard curve, and that's no problem.
Okay, great, thanks. And the other question I had, you mentioned protein-protein interactions. So do people do immunoprecipitation and then they give you the immunoprecipitate and you're able to distinguish the two proteins from the antibody, or like,
Yeah,
I'm curious.
That's just depend on, and we do, I do not do the immunoprecipitation. So, and, if you do that and you generate the complex of that, sometimes we can work on the cross-linking of the two proteins and to link them together. That's easier. If it's non-covalent interactions, that's just really depend on how tightly they interact with each other. So we have worked, with just non-covalent interaction before, and it just it worked, but it's really depend on how tightly the non-covalent interaction is. Yeah, we have seen dimers or trimers of peptide proteins.
Okay, great. Thank you so much.
All right, I'm going to turn it over now to John.
Okay. All right, so sharing. So, okay, I think I'm sharing. Am I sharing? I'm not sharing, am I?
Not yet.
It told me I am.
There we go.
All right. So in essence, swimming provides a heavy speciality in activity. And what the rest of the core facilities in PSC and NSC and the ABC core, they provide essentially the rudimentary molecular biology activity that many of you may enjoy, some instrumentation in your lab. Otherwise, you may have to outsource some of your activity. And so what we provide essentially predominantly is the capability to utilize hands-on any kind of instrument that you would normally have within a core facility, within a regular facility. And as you can see, some of them are cost, some of them are free of charge. If there's no little cost across here, it's all user activity and user dependent. We don't charge anything for these activities. But here we have essentially on the website, which is biotech.gsu.edu/core, will get you there, gives you a background in what we are able to do with instrumentation and in both location in NSC and or PSC. So here, for example, is in PSC by location. These are all the different equipment that are available to you to use as you will. And we will happily train you and your staff on how to use any and all of these facilities. And that's the goal of this avenue of the core, is that we provide a hands-on ability to utilize and prepare all these activities at cost for you to use. We do have professional staff, technical staff, who are available. This is actually an old one. It should be a picture of Jesse Gardner, who was hired in the last spring. And each of these members have a capability. Sandy is involved in cytometry, genomics, qPCR, etc. Sonia is genomics and some flow cytometry, and nanostring, as is Jesse, and Q in protein aspects. All of these are experts in their field, and they are there to help you both utilize the resources and also at times allow you to go ahead and avail our core facility as a service. So we prepare, we give you hopefully the full gambit of instrumentation to utilize as a regular molecular biology core, as well as the services and this expertise to go ahead, for example, and with Sandy, go ahead and use any of these cytometry equipment, from a mobile unit to the heavily used LRS Fortessa, which is about 4 lasers and 8 filters, can handle about 13 different dyes, to actually going ahead and sorting up in the 6th floor. So we provide a range of expertise from fundamental use by yourselves to a service that Sandy provides in certain areas of that activity. So this arm of the core is designed to give you all aspects of potential use within the realm of molecular biology. So if I just go through very quickly, this is what the core does. These are the people in the core. What do? We provide expertise and service, but we also provide training for all your incoming staff, students, and everyone else to work in the PSC and in the core facilities over in NSC. You have to have access by swipe card, and we are the gatekeepers. We provide that aspect as well as training on general use of equipment within generalized areas of the core facilities. So for example, currently we have video on how to utilize distinct areas or distinct instruments like autoclaves, film developer, high-speed centrifuges, and so on and so forth. Generic, but required if you want to utilize core facilities. We also provide expertise in certain areas, but we also monitor and regulate access. We oversee and maintain the instrumentation within the core facilities, and we repair if possible. We promote and advocate departmental grants and tours, and essentially odds and sods. We are the go-to people to provide equipment moving in the biology, chemistry areas, and helping repair, replace equipment, even if it's not particular, non-core. Our revenue stream, to show you the size of the business in essence. Last year, we only did about 90,000. That's with COVID. The previous year, we were 130,000 revenue. And this is use of the instrumentation and availing yourself of aspects therein. Now, that's the revenue. It's broken down by instrumentation, by sequencing, by flow cytometry, by ion tolerant sequencing, by plate readers, real-time PCR, and so on and so forth. And this is done over the various years from 2010, 2019. This is how it breaks down in essence. We began heavily as a sequencing module and then expanded to cover a large variety of different pieces of equipment. We have an active and increasing activity in flow cytometry. As I said, both instruments are able to handle between 2 to 4 dyes, 2 to 4 lasers, and about 13 to 8 different dye activities. We cell sort. We also, while we're not rich and we are underfunded, we do also purchase equipment in the mid-range, so about $50,000. negotiate with different faculty who want to buy certain pieces of equipment. We also invest in student technology fee proposals. And each year or so, we try and invest in purchasing equipment that everyone uses. This would be a Keon's fluorescence microscope. Then we went ahead and did DNA sequencing next generation with trade-in. And this is the low-end genomic sequencer. It's equivalent essentially to the MiSeq to some extent, but it's far more rapid, a little bit more costly, but you get the turnaround more quickly. Last year, year before last, we invested in the nanostring sprint profiler. And this instrument here, whoops, didn't show. Instrument here essentially utilizes barcodes of dyes, which you purchase from the company. And then you interrogate anything from proteins to nucleic acids to try and work out essentially their concentration and their presence in different areas of the cell, and so on and so forth. And all these people around are actually involved in purchasing the equipment. Dr. Gaucher was the chair of department biology 2 years ago, and he was heavily involved in getting this new instrumentation. We're not rich, but we're flexible. Aside from all the equipment that you have available to you, the idea of the core is also to respond to activities. And so when the SARS-CoV pandemic came along, Julie Hill and myself set up a CLIA certified COVID-19 PCR test. And a majority of instrumentation came from the core facility and allowed it to, because of the size of the facility, we were able to commandeer certain units and provide this for the testing lab for the last 6 to 9 months. Along with Dan Diacampo's help in synthesizing all the swabs, we were able to go ahead and test a variety of features. Last 2 weeks, COVID storage. We emptied out 4 -80 freezers and other storage to provide a vaccine capability for the university to be able to store vaccines. Hopefully they will arrive in the next couple of days and be administered to faculty and staff in the days thereafter. So this is just an overview. It really is dependent upon you going around, if you can, the core facility website, finding out what we have, asking questions of the different staff and what they can do and to help you. But we cover a variety of instrumentation. We have some speciality, but really it's generalized. So at that stage, I'll stop. You have all the information there on the website to communicate, but I'll stop there for questions. Pardon me.
Thanks, John. And I love to hear about emptied out freezers for vaccines.
It was a rapid process.
I'm like, I will give away all of my freezer space if I can get vaccinated.
Well,
What's exciting is that you said in a couple of days, well, the GSU will, have the vaccines.
it's been handled by Tim Denning and Brenda Chapman. And if EJ Asanobi, the student medical officer,
Oh,
she's actually in control of giving out the vaccines. We're expecting about 2,000 to 3,000 this week, again, depending upon supplies.
So that will serve to GSU community only?
The idea is GSU community gets priority, and that's the idea of filtering out the vaccine capabilities. And she essentially, well, they will tell you when you are able to, but the underlying, proposal was that the faculty and staff and students who are here on campus get priority.
I love to hear that.
It could be students, staff, and faculty. That's at least the overriding principle.
So any questions for John?
Yes, I have a question. Thank you, John, for the presentation. I was wondering about the post-hoc analysis of all the data that comes out, for example, out of next-gen sequencing. Do you provide that?
We are we are beginning to provide that. We have access to an Advil resource at Georgia Tech, who are committed to go ahead and analyze the data. previously, our technician was not able to analyze data beyond a rudimentary activity. But we are branching out. Jesse was hired last spring for that purpose. He's a master's student who is completely fascinated by analysis of sequence, and therefore he was hired for that purpose, to both make it move into genomics and also to provide analysis thereafter. And we're also collaborating with University of Augusta, University of Georgia, in the COVID response. We collaborate through there, and we're doing the sequence analysis with them, and we will definitely learn lots of ways to analyze both Illumina sequence and also ion torrent sequence. It's an ongoing project, so we're not quite there yet, but we will have, I'm hoping by the end of the spring, a suitable mechanism to have a one-on-one activity. But right now we're working with Advil over in Georgia Tech for analysis.
Okay, thank you. And how about single cell transcriptome sequencing? Is that something that you do?
Okay, so yes, we do. And that's part of the nanostring, is the preferred model right now. It provides for far more specific analysis, and those are around $300 per run. If you pay for all the dyes, we provide full service for about $400, and that would go. If you go to the website, you'll find that at the website. Let me show you the website. Okay, so this is NanoString. This is the website. This is what you would provide. You communicate with the company for buying the dyes. Custom panels are between probably about 1,000 to about 3,000, or you can buy them for about a few $100s, $500 per panel. You buy the cartridges and then go ahead and run the samples, and Sonia and or Jesse will help you with that activity. If you want to go at the level of, of next-gen genomics, then Jesse's the person who would talk to. And this is, right now we haven't got transcriptomics up and running, but we have a call for pricing for genomic sequencing, and he will definitely work with you on that pricing.
Okay, thank you very much. That's great.
I'll also just echo on to that for JP that through the IINT and basically our bioinformatics support team, there are clusters of available software for, you know, your standard kind of pipelines of bow tie, et cetera, et cetera.
Yeah.
And if you need contact information, I can give that to you. The primary person is Saranga.
Okay, thank you. That would be great. Thanks, Dan.
Any priorities, John, for what you see as new equipment purchases? Is there anything that the core is particularly looking to add?
Well, okay, like Siming suggested, all our high-end instrumentation is a little bit older, and our revenue stream is able to handle about the purchase of one or two items at around 45 to 100,000. Anything more than that is requires a lot of collaboration with both departments and individual researchers. So normally last year we bought some spectrophotometers. This year we're buying some high-end PCR, RT-PCR units. So our instrumentation purchase is at this stage mid to high-end fundamental usage. We haven't got any specialized equipment on the docket. The last one actually was NanoString, and that was purchased because the department had a circuit of $100,000 to spend. We haven't got any direction right now with respect to what we need to buy. We really need to maintain our flow cytometry and our cytometric cell sorting. And that's where I think we will be going in the near future for relatively high-end activities.
Okay, great. Well, let me turn it over to Kwanchai Chen for the imaging core facility.
Okay, I will be sharing my slides here. can you see my slides?
Yes.
Actually, let's go to the top. So my name is Quantai Chen, and I work at the Imaging Core facility. We are a specialized facility. We're a small unit, mostly focused on the imaging. And we provide advanced light and electron microscopy imaging-based tools to researchers to different departments and also external users. You can find us by searching Imaging Core Facility GSU easily. You can find all the basic information about instrumentation, training requirement, and reservation, and also contact informations. So as a shared facility, we have a list of shared equipment. I will briefly introduce a few of them listed here. We have 2 laser scanning confocal microscopes in PSC 839. They are LSM 700 and LSM 780. Both of them are from Zeiss. And we also have a scanning electron microscope in PSC 835. It's from T-Scan, and it equipped with elemental analysis capability, EDEX. We also have wide field microscopes, one upright and one inverted in 835. So confocal is widely used for imaging many different samples, from cell monolayers to thick tissue sections, and with fluorescence label or endogenous fluorescence signal. Users can be trained to acquire their own image on the systems. Since I did not involve in acquire any of this image, I will leave this to you to explore. You can go to our website and get into the image and gallery, and if any of the image grab your attention, you can click on it and you can find more specific information of which lab acquire this image and what specific information of this image are. And for SEM, I do the operation imaging for the users, and it's very useful in many different fields. So for material science application, I list a few examples here. Imaging the gold nanospheres and gold nanorods. Also, chemistry professor Dr. Shamsi, they use SEM to image their monolithic columns to optimize and evaluate their reactions in the capillary column. And Dr. Ningfang's lab in chemistry use SEM for imaging their 2D material, molybdenum sulfide, and they use this material for study the photocatalytic reactions in different regions. The image on the bottom right shows the tin nanoparticle being imaged by the SEM. The particle size is ranged from 20 nanometer to 140 nanometer. And this actually marks the resolution of the Vega 3 SEM that we have. So we can image down to about 20 nanometer if the sample is prepared very, very well. SEM also find a lot of application in life science. Neuroscience professor Dr. Charles Derby used SEM to image the white shrimp claw and the chemosensory organ on the teeth of the shrimp claw. So you can see on the top left, figure B, it shows clearly the pore structure on the tip under high magnification, and they can see all these details of the teeth development. They recently published their results in the journal Biological Bulletin. SEM can be used to image allergenic pollens, hairs, and also like BIs. On the bottom right, you can see with a high magnification, you can see the micro length of the BIs and all the detailed structures and the surface morphology. IBMS Professor Dr. Chris Basler's lab used STM to image their Ebola virus inclusion body, and this inclusion body was labeled with the magnetic beads that coated with antibody to specifically target the inclusion body. STM also find a lot of application in other fields. One postdoc in Dr. Basler's lab used STM to study the filtration efficiency of different masks. So the use of STM to image cross mask and also N95 mask with tested with in the lab, we were able to visualize individual SARS-CoV-2 viral particles on the surface, and we can tell that N95 give much better filtration efficiency compared to the cross mask. Other sample like STM can be used for both conductive, excuse me, conductive materials or non-conductive materials. So with backscattering detector, we're able to image uncoated sample, paper sample, and with coating applied, we can image all the details of the composition. On the top right, the 3D printed bunny was printed in Dr. Sudong Lei's lab in physics department. They built their own 3D printer and tried to use SEM to evaluate their printer quality, or the print quality. SEM also find use in geoscience. Professor Dr. Dan Campo, also our associate dean, use STM and EDEX function to study the mineral sample, and you can use the EDEX for the elemental analysis to figure out different composition in the mineral. So in the middle part here, in the flat area, these are the aluminum-rich clay, and on the surrounding, these contain less aluminum. Professor Dr. Gangli Wang used SEM and also EDEX to map their crystals fabricated in the lab, and you can tell different composition in the crystal that they made. So most of people know us because of the confocal. However, we also have wide field microscopes. They are useful for certain samples if it's not too thick, and there's different imaging capability available on the wide field microscopes: epifluorescence, bright field, phase contrast, and differential interference contrast, dark field, and polarized light microscopy. So for a relatively thin sample, multicolor imaging is very accessible. So on the left side, it shows the BPAE cell with 3 fluorophore labeled being imaged on the epifluorescence. And on the right side, the same cell, their actin structure can be visualized, and the details can be reviewed by the super resolution radical fluctuation on the epifluorescence microscope. Bright field is another very useful tool for imaging chemically stained samples, especially in histology or pathology sample. You can either use a low magnification to image a larger field of view of a sample, or you can use a high magnification and scan through the whole slide to get the higher magnification of the whole slide view. So we have a high resolution imaging camera, which has about 20 megapixel. And if you compare that to 4K resolution, it's near 10K resolution for the image that we acquire for each of them. And what's more, the software is able to do the live imaging staging for the whole slide scan. So you can scan through the whole slide and get a high resolution image of the tissue sample. Dark field and DIC on the microscope can be used to image single particles. In this case, 100 nanometer gold nanospheres are both imaged under dark field and DIC. You can clearly see the slight difference of individual particle, which cannot be seen in the ensemble measurement like UV-Vis. That's all that I have. And about all the other questions, you can either find the information from our website or you can contact me directly through my email. Thank you.
Great, thank you, Kwanchai. How small is this bunny?
Here is a scale bar. It's 10 micron, and actually it's smaller than human hair.
That's amazing.
What surprised me was that they actually built their own 3D printer. So at the beginning, I thought they bought a printer and then tried to print out, but they actually built their own printer.
Yeah, that's pretty impressive that you could print something that small with that much detail on it. Cool. I'll open it up for any questions. Oh, do you want to say something about the recharge rates?
Yeah, so currently we charge $20 per hour for 700 and $25 per hour for 780 for unassisted imaging. So users can be trained and then come in to acquire image. And there's also option that if you only use one time or two times, it can be the sample can be handled by our confocal technician, Claudia. For the SEM, we charge $50 per hour. And wide field, if you use it extensively and use the new cameras, it will be $10 per hour.
Great. And are there priorities that the core facility has for areas that they want to expand in terms of new instrumentation?
Yeah, so the confocal that we have are both old and they need to be replaced at some point. And I'm looking into possible funding source to upgrade the confocal in the future. And also for the wide field microscope, so tiling function is very useful for whole slide scanning. However, this is done by just manual scanning. So I'm hoping to get a motorized stage that doesn't need to be very high resolution. As long as it can scan through, it can acquire the image easily.
All right.
I had a question. Sorry, Dan, to interrupt you.
Hi, Jessica.
Hi. Yeah, sorry I don't have my video on. It's too early here.
No problem.
That was wonderful, Kwansai. I'm really interested in all of the imaging methodology you guys have in the imaging core. I'm a new faculty member in the Neuroscience Institute. The motorized stage would be super helpful if you can get that. I was also curious, I've heard rumors about a light sheet microscope. Would that be something that would be through your core or somewhere else? I can't remember.
I believe there's one light sheet in biology department in individual lab.
Okay, an individual lab. Okay, not the core. That makes sense. And then I also had another question for the SEM. Do you offer any help with sample preparation or the ultra thin slicing?
We do basic sample prep like conductive coating And we
Okay.
also have shared equipment in the facility that you can use for if you need to do the section.
Yeah, the sectioning, definitely. That's good to know. Thank you. I think that was all of my questions. So thank you very much.
You're welcome.
Any other questions for our panelists as we're approaching the end of our scheduled time?
Yeah, thank you for the presentation, Kansai. I had a question about deconvolution for your confocal microscopes. Can you reconstitute 3D images from your image?
For the confocal image,
Yes.
there's immerse software and workstation. I think for this you can contact them for more detail. There's several groups share that, and they have their own schedule. It's located in the core facility, but Dan is in charge of that.
Okay, thanks. And I was going to ask about inverted microscopes for calcium imaging. Is there who would have that as a core?
We do have an old microscope for calcium imaging, but I will need to. I can contact you on that maybe later for more specific details.
Okay, sounds good.
It's located in an individual lab right now.
Okay.
So JP,
we also have a calcium imaging over in the science annex. If you want to use that, it's an imager that can handle calcium and is routinely used by by Vincent Rader's lab and others over in NSC.
NSC. Okay, thank you.
I'll just echo onto that, JP. So it depends on whether you're, prioritizing the use of genetically encoded calcium indicators versus like Fura or.
Yeah, we were going to do Fura ratiometric.
Yeah, so if you're going to do ratiometric imaging, my advice is to follow up, as John suggested, with it probably is under Liana Artinian. And she has expertise with Fura-based imaging.
Awesome. Great. Thank you. Thank you both.
All right. As I said, if the panelists are willing to share their slide decks with me via email, I'll make those resources available. And I just want to thank, John, Siming, and Kwangchai for giving their time and preparing this information, as well as the participants for logging in. Hopefully you found this useful. I did.
Thank you. It was very useful.
Please call if you need help.
All right.
Thank you.
Happy to give it.
Take care, everyone.
Thank you. Bye.
Cheers.
Bye-bye.